which is change of species. So this is where you have one species diverge into a different species or two species or what have you splitting. We'll talk about that next week. That'll be jumped into next week. Okay, so far we have covered natural selection, which was the first. So natural selection. And this was by Darwin in 1859, when Darwin published this one. We've already last time described, the three different flavors of natural selection, which would be directional selection, stabilizing selection, diversifying or destructive selection. But anyway, the whole notion of natural selection is that you have a population with variation, that some individuals have an advantage from their variation and some have a disadvantage. And that variation is heritable. And so the ones that have the advantage have more offspring and pass that advantageous trait to their offspring. So the next population kind of is favored for those that have the advantageous traits. That's quite a bit different from what Lamarck, Jean Lamarck had proposed, right, which was acquired traits, whereas Jean Lamarck suggested that, organisms have some sort of an environmental strain or some sort of physical strain, like the giraffes were reaching up high into the trees, and that strain on their neck reaching for the leaves will be passed to the offspring, and the offspring will basically respond by having a longer neck to be able to reach higher in the trees, and so on, right? So it is kind of interesting that Lamarck used the giraffes as an example. Darwin used the giraffes as an example. Among the giraffe population, there's tall ones and short ones. Tall ones get more food, grow larger, have more babies, and the short ones lose out, and the population basically gets taller over time, right? Make sense? It's the same result, but it's a different mechanism. So be prepared to compare those two mechanisms, right, on the test. So be prepared to, like, maybe explain, for example, the giraffe story as Lamarck might propose a mechanism, and be prepared to explain the giraffe story as, like, Darwin might propose it, right? But now we know the real answer, or at least for today, what the answer is for the giraffe story, and that is it's actually a sexual selection deal. It's not about resources, food resources, it's about mating resources. So we know that now what happens is the male giraffes are kind of lurking or stalking the outside of the female herd and looking for opportunities to be the male that has the opportunity to mate. And among the males, they'll basically compete for that opportunity to be kind of like the male in the wings, right? And so they'll fight with their height and their neck and their horns, and the male that wins gets the opportunity to reproduce. The male that doesn't win loses out, right? So that's basically what sexual selection is. It's like natural selection. It's just that the limited resource is mating opportunities, right? So Darwin actually wrote a book about sexual selection. He wrote about sexual selection. He didn't know that the giraffes were an example of this because he never saw a giraffe live or studied them. That's what the big kind of joke was, that it took 150 years for elementary school kids to be told the truth on giraffes, right? But Darwin did write a book about sexual selection in 1860s. So he realized that sexual selection was an alternative mechanism to natural selection, in that organisms can have, like the giraffe, this kind of really bizarre kind of feature, which is a long difficulty drinking water and have to have high blood pressure, large heart, etc. But it was all about essentially mating up. And there's other examples as well. Like, think of like a moose or deer that have these large antlers. I mean, male deer that are around the bucks in the North State area with the large antlers, that's not really necessarily for fighting off predators or for defending themselves. It's for sparring with other male deer for being the dominant male in the area and for attracting females, right? So this is a sexual selection type of situation. But a tremendous amount of energy is put into growing these, long antlers every season, and it also puts a lot of difficulty on those bucks because of the antlers can get caught and snagged in brush. I've found young bucks caught in fences, and so sexual selection is an interesting story because you have organisms that have seemingly bizarre traits, like birds, peacocks with their big plumage, right? You'd think that would attract predators versus having camouflage. But it's all about, mating opportunities and being able to pass the gene along, okay? So, probably, here's the test question. Here's just all I can say what it is, right? Is, this giraffe story giraffes have long necks. What was Lamarck's mechanism for the giraffes? What was Darwin's mechanism for the giraffes? And what is the current today understood or agreed upon mechanism for why giraffes have long necks? And so you'll have to talk about Lamarck's acquired traits as regards to giraffes' necks. Acquired traits, of course, means there's some sort of strain or physiological transfer of traits. Darwin was more about a population variation, and the ones with the favored trait reproduce, and the ones without it don't. And then currently, sexual selection is traits are for a mating advantage, regarding success. Okay. So, any questions on that? I'm just telling you about that because in the past, when I've given that test question, people kind of jumble it up or don't give me enough information. So it's important that you kind of sort those out and you understand the difference between like acquired traits, which were not correct, natural selection, which is correct, but this isn't the reason why giraffes have long necks. It's sexual selection. And even though Darwin knew about sexual selection, he didn't apply it to giraffes because he didn't study them. Okay, moving on. There are a couple more mechanisms for microevolution that I want to talk about. Namely, one that is called genetic drift. In the 1930s, and I'm not going to hold you responsible for knowing names, but possibly in the question, I would present the name as a way to kind of jar your memory. But, so don't worry about memorizing names, but there are two population ecologists that came up with this in the 1930s. So yeah, we're talking like a full, 90 years after Darwin, right? Or not 90, but 70 years after Darwin. They proposed, Sewell and Wright proposed that you can have drastic changes in population traits based on small population sizes and non-random gene transfer. Let me go ahead and explain what this means. The whole idea here of genetic drift is that when populations are large, populations are quite big. Let me get up that image up here, real quick here. I forgot to hold my PowerPoint here. Just a second. All right. So the idea of what's called genetic drift is when populations are large, typically traits or alleles will stay the same from generation to generation. So the other term that's used to describe genetic drift, or the mechanism for genetic drift, is what's called the bottleneck effect. So genetic drift is the thing that you need to know. One mechanism is what's called the bottleneck effect. And why it's called that is oftentimes in population demographics, they'll show populations kind of like a bottle on its side, and then the population squeezing down into a small opening, a small neck. But you can think of like if I had two alternate alleles, like let's look at those. One example would be like those the pocket mice that we had as an example for disruptive selection. If I had like a light coat color and a dark coat color, I mean these are like jelly beans in a bottle, but there's some blue ones and some yellow ones. So let's think of like a dark coat, like a blue coat, and a yellow coat, and a lighter coat. Typically, if the population is large, like in the tens of thousands of individuals, the frequency of those alleles would stay the same from generation to generation. But if I took just a small portion of those individuals and only allowed them to reproduce, so if I randomly only let like 10 individuals reproduce, there's a good chance that I'm no longer going to maintain that allele frequency. So let me go ahead and show you an example of this. There's some cool simulations out there for genetic drift. So I'm going to go ahead and set this simulation up where there's two alleles. Let's go ahead and say it's like there's a dark coat allele for the pocket mice, and there's a light coat allele for pocket mice, right? I'm going to set my population size as 10,000 individuals. So 10,000 is a pretty good population size for some number of generations. I mean, it's not really a super large population size. I mean, we know that Reading has over 90,000 people here. So 10,000 is kind of a modest population size. You could think of like there's probably like in the deer population might be somewhere around 10,000, like out in the Trinity and Siskiyou County kind of area. But anyway, here we go. Check this out. So I hit evolve, and the allele frequency is 50%. And what happens is, this is kind of like flipping a coin. You think about like the exercise of flipping a coin. If I were to flip a coin, and I'd say there's two alleles, there's a heads allele and a tails allele. The frequency or the probability that I'm going to get heads or tails is 50%, right? We have a 50% chance of getting heads, 50% chance of getting tails, and that's like on a per generation basis. The difference here is the next generation is affected by the frequency that we get from the prior generation. So if I were to flip a coin 10,000 times, you would expect 5,000 heads and 5,000 tails. But if I got like 4,500 heads, if I got like 4,500 heads and 5,500 tails, you wouldn't think there's any trickery or anything. You'd think, okay, well that's expected from like random. I might just get a few more heads and tails because of random chance, right? That's what the mechanism behind genetic drift is it's random events that happen in frequency, right? Even though there's a 50-50 frequency of dark and light alleles, the chances of picking those have some variation. So what you can see is we are slightly drifting up in the frequency of, let's say, the dark allele was the number here. So we're kind of slightly drifting up, and that's why it's called genetic drift, because there's no selection going on here. There's no resources or gating thing. It's just all about just random chance when you have the frequency of alleles altering over time. Okay, so this is with a relatively large population, 10,000 individuals. You can see we've now drifted up to 60%, right? So that's pretty good. I mean, we've altered the allele frequencies by 10% just because of random changes that happen now over 280 generations. Okay? So I'm going to go ahead and let's see how we stop this. Oh yeah. Okay. So what I'm going to do now is I'm going to reduce the population size by an order of magnitude. I'm going to decimate the population. And when you decimate a population, you basically move a zero over. Okay? I'm going to take it from 10,000 to 1,000 individuals. So I've decimated the population. The population is now 1/10. I've killed off 90%. I'm retaining 10%, right? Check this out. So here's 1,000 individuals. And you can see that it's beginning to kind of deviate much sooner. So now within 25, 30 generations, we're now, we've deviated by 10%. It doesn't really matter whether it goes above or below the 50% line. The fact that we've changed the population significantly from its original point. Okay, so this kind of, it's drifting around. So this is why it's called genetic drift. All right, so I mean, we could watch this go on and on. Let's go ahead and do this. Let's take it down to 100 individuals. So I'm decimating it again. It's kind of like when a wildfire happens, right? Wildfire happens, and this, the squirrel population is kind of hammered, right? Or a disease happens. All right, 100 individuals. Now you can see that it's more wildly fluctuating, right? So. The other way, another example might be to think about is the Galapagos tortoises. Remember I mentioned that when Darwin saw the tortoises on the Galapagos Islands, that the locals there knew which island the tortoise was from? And the reason was because some of them had a higher dome, and some of them had like a saddle shell, some of them had kind of a flare behind the tortoise's neck. Each island kind of had its own unique trait or flavor of tortoise shell. And it's likely with tortoises that it had nothing to do with any kind of a resource advantage. It probably was just those tortoises were isolated on their own little island with a small population. And they ended up kind of looking different because they were not able to interbreed with tortoises on other islands. Check it out. We've already deviated now at a population of 100. We've deviated it down to, it's gone this way, and it's gone this way, and now we're up to, 80% frequency. So we've drastically changed the allele frequencies just because of random chance. I'm going to take it down to 10. Check this out. So, huge fluctuations in allele frequencies based on just random chance, and I'm waiting for this to happen. But when it hits 1 or 0, what happens basically is, oops, there you go. So now what happened is, let's imagine that this was the frequency of the dark allele. We basically lost it, and what that means is that allele disappeared in the population based on just random chance when populations are at small sizes. So, the mechanisms you should know for this mechanisms are what's called the bottleneck effect. Bottleneck effect is probably the most widely cited mechanism for how this happens. What that means is the population reduces from a very, very large number to a very small number, and as a result, random disproportionate representatives of the population reproduce and alter the allele frequency because of population size. The other mechanism that can happen is what's called founder effect. It's the same result, But in a founder effect, what happens is, let's imagine like you have organisms colonizing an island. Here's the Hawaiian Islands. And in the Hawaiian Islands, the big island, Hawaii, is the youngest. It's like 1.5 million years old. In fact, it's still growing, right? It still has volcanic activity. But as you go out to the northwest, Maui, Oahu, Kauai, the islands are older and older and older. Like Maui is like 3 million years old, and Oahu and Kauai are like 5 and 10 million years old. And so what happened is as a new island is formed, it gets colonized by individuals from other islands. And usually colonization happens by just a few individuals, like, out of a population of maybe 10,000 flies or whatever that occur on Kauai, maybe a dozen get carried by the wind, or their eggs get carried by a bird or whatever to Hawaii, right? That's called a founder effect, where you have a small population that establishes a new population. And the end result is the same. It's a small population size. It's based on a small population. So, small populations are subject to genetic drift. Large populations tend to remain relatively constant. So check this out. We have, one example would be like elephant seals on the California coast. So, 120 years ago. I guess let's put it this way. In the 1850s and 60s, right? The only oil that they had to lubricate machinery, like if you wanted to lubricate your locomotive train or lubricate your machine that is manufacturing, that was all lubricated using whale oil. So whale oil was the standard, machine oil lubricant back like in the 1800s, 1860s, 1880s, so on, 1900. So as a result, whales were over-harvested. They were basically annihilated in the oceans, especially off our California coast. Trinidad was the big whaling center off California, for that time. When the whales were overhunted, the whalers basically were looking for alternative sources of blubber, and they would basically target the elephant seals, because the elephant seals would haul up on the beaches in certain areas, like down by Monterey, and down all the way down to San Diego. They haul up on beaches to mate in April or so, April and May, and all the whalers had to do was just go out there and just shoot them, club them, take them away, right? So it was super easy to harvest the elephant seals. So the population, I think the estimate was there was possibly as many as 100,000 elephant seals that occurred from Baja California and San Diego all the way up to British Columbia, Victoria. And then by 1910, the early part of 1900s, there was maybe as few as 100 of them. So basically the elephant seal population had that same result as I showed in the simulation, is the population bottleneck, or in this case, it was population bottleneck, where they were overhunted or they were exterminated from, potentially 100,000 individuals down to maybe 100. You got to imagine that if we had a gene pool that had a number of alleles, we probably don't have a representative current living example of what the genetic diversity was. Because that small population will not only alter, this population has, a 33% allele frequency for A, B, and C alleles. Just use your beta alleles. And then now we have, 75% C, 25% A, and allele B is extinct, right? And what happens now is this population is the one that is the reproducing individuals to make the next population. So today, I think they estimate there's like maybe 10,000 elephant seals that have rebound, have kind of come back. And, but it's pretty clear that the genetic makeup of the current population is quite different from what the original one was 150 years ago. So these are very important. So on the exam, and again, I do this every semester now. I kind of say, this is what's on the test. And then when I'm reading the test, I'm kind of baffled as to why people space out. But anyway, if you see an example of, in the example in the question, where a population goes from a quite large size down to a very, very small size, like you move a decimal over a couple places, right? You go from 100,000 down to 1,000, which is the decimal twice, right? You're immediately going to think, oh, this is genetic drift. And the mechanism you should choose is, if it's because they were killed off, it's a bottleneck. If it's because a small population migrated to a new location, it's a founder effect, right? But either way, these are both what causes genetic drift. Got it? All kinds of examples of this. Think of like condors, right? Like the California condor. The estimates were like before settlers showed up in California, like in the early 1800s. Estimates were there was probably like tens of thousands of condors all up and down California Central Valley. And condors' primary food was, they're like a vulture, right? Their primary food was like dead elk, dead deer, and large mammals that basically died is what California condor eats. And there were large herds of elk and deer all through the valley. So when the settlers showed up, not only did they kind of wipe out a lot of the large deer herds and elk herds, but also lead poisoning led to the California condor being reduced down to probably less than 10 individuals. Like they had them down to 10 condors. And I think they took most of those into a zoo just to kind of keep them alive back in the 70s. Well, the condor program has, successfully reproduced condors in the zoo, and then they released them back into the wild, and now with banning lead ammunition and, kind of conservation efforts, there is now probably several hundred condors, maybe even as many as 1,000 condors around California. But imagine that, going from 10,000 down to 10, and then from those 10, you reproduce, certainly a lot of inbreeding is going to be happening at that few individuals. But now the population is probably quite different genetically, from what existed 150 years ago. So California condors would be a great example. If I were to put that on the test, but it won't be that. But that's just an example. Okay, so again, a hint. If you see an example where you go from a large population to a small population, and there's a little chart or graph or something that tells you that in the question that the population goes from 100,000 down to 1,000, that's your answer, right? And then your mechanism will be based on whether there was a die-off or termination, or whether there was migration, right? Okay. Just telling you, we're moving on. Okay, two more mechanisms I want to touch on, but I think one of them I'm going to mention, but I'm going to come back to it later. I'm going to come back to it next week. And then the other one I'll touch on, and I have a paper to show you on the Canvas site to look at. But anyway, okay, one of them is mutation. So the important thing about mutation, and this is basically where there's changes in the DNA code. And we can kind of think about this. I'm not going to give you a name on this one, but this one, basically the 1950s is when the DNA code was, and the DNA structure was described by Watson, Crick, and Franklin, described the structure of chromosomal DNA, and then soon thereafter, we had the realization that genes can be changed by mistakes when the genes are coded. So when the DNA replicates, there'll be some rate of errors that are coded when the DNA replicates. We call this like a natural background mutation. Every time your cells divide, you have about 30,000 ATGC letter codes that get changed or altered because of just a mistake in the code translation. So anyway, that's mutation. Mutation is where new alleles come from. So if you have a mutation in the DNA code, there will basically be an altered allele possibility happening. So I'm going to go ahead and just leave that there. We're going to revisit that with macroevolution because there is a whole genetic species concept that I want to talk about. And the genetic species concept uses mutation rate and DNA change as kind of its basis. So we'll leave that there. Finally, this is a pretty relatively new one, and it's called horizontal gene transfer. Horizontal gene transfer, I'm going to say that this probably was made clear in about the 2000s. So in the last 20 years, this has become kind of clear that this is happening. The reason why horizontal gene transfer became known is when genome sequencing became possible. So genome sequencing became possible like in the 1990s. You know, the human genome was pretty much sequenced and done by about 2000. And at the same time, they sequenced like the dog and tobacco plants and fruit flies. Essentially, they did gene sequencing on animals and plants that were kind of common research organisms, on humans also. And what they found was there were regions of genes that were shared between organisms that weren't related at all. Like they found that well, I mean, here's an example. I'm not going to give you a speculative one, I'll give you a real one. Why not just give you an actual example here? And horizontal gene transfer. So anyway, here, check it out. I have on your Canvas site, go back here, on lecture 2.3 is an article. It's a short article, but you probably want to check it out on horizontal gene transfer. But check this out. Gene transfer between species is surprisingly common. So here is the story. Remember last time we had the protist Trichomonas vaginalis? This was the one that causes the sexually transmitted protist, right? It's the reason why you can't return underwear after you put them on. Anyway, the Trichomonas vaginalis, which causes the sexually transmitted protist, thrives in your, basically in men's urethra or in women's vaginal cavity, right? So essentially it lives in your vaginal cavity or urethra. There is some problems with living in these environments. You know, the pH environment, for example, in the urethra is kind of acidic, and the pH in the other environments, you know, so basically it's kind of a challenging environmental condition. So anyway, when they did a gene sequence of Trichomonas, they found that it shared a huge chunk of genome. Let me see if I can find it here. They have an example. Oh, I thought they had a picture. Anyway, they found that it. Let me pull up a picture of Trichomonas. when they did a genetic sequence of Trichomonas vaginalis, they found that it had a significant genome sequence, like 2,000 base pairs, that were almost identical to a bacteria that causes urinary tract infections, right? So here's a bacteria that causes bladder infections or urinary infections, UTIs. And so obviously that bacteria is adapted to living in the urinary tract. They found that Trichomonas had a large chunk of genes that also occur in this bacteria. Now Trichomonas is a protist, right? It's a flagellated protist, eukaryote. The bacteria is a prokaryote. These things could not hybridize, right? There's no possible way they hybridized, except horizontal gene transfer sort of is a mechanism of hybridization. It's where when two species are closely aligned, one will kind of absorb the other one or steal its genome. So it's likely at some point in time, or various times, the association between Trichomonas and this bacteria that causes UTIs resulted in probably Trichomonas absorbed the bacteria or consumed it or something like that, and a chunk of genome was borrowed and spliced into its genome, and those genes enable Trichomonas to live in that habitat, where the bacteria lives. So Trichomonas, whose closest relatives are not urogenital parasites, was able to occupy that new habitat by adopting the genes of the bacteria that already lived there. And so you're thinking, wow, that's pretty far out, man. That's like, that's a crazy example. But there are all kinds of examples. When the human genome was sequenced, they found all kinds of bacteria gene sequences and fungal gene sequences spliced into the human genome. So it's becoming quite clear in the last 20 years that genes can be transferred between very different types of species. And in some cases, the species that gets the gene can express them and use those as adaptive traits. And so this is kind of a forefront article. And you can see that, to you, this might be a long time ago, 2007, but that's pretty far out. In the last 20 years, this has kind of been something that is striking. So if this is only like maybe 1.5 pages, I would recommend that you kind of just scan that or check it out, because I'll probably have an example on the test of a horizontal gene transfer, and it might be from this article. It might be something that's mentioned in the article there, so you kind of get an idea of explaining it. But basically, your answer would be horizontal gene transfer is where genes from very, very different species, like different kingdoms, can be adopted or taken and incorporated into the primary organism's genome and expressed. And that is like a crazy hybridization, right? You're thinking like this is like science fiction, but it's pretty neat. Okay, so that being said, here is our list of microevolutionary mechanisms. So was Darwin wrong? No. Darwin just knew about these things, right? He didn't know about any of this because, the understanding wasn't there yet. So what we're doing basically is we're saying it starts here, but we have incorporated other explanations into the microevolutionary concept or theory here that result in change of populations over time. We're going to revisit mutation when we talk about species definitions next week. Okay, so I'll come back to that. All right, so what I want to do, we have like maybe 5 minutes remaining here, is how do we make the connection between microevolution and macroevolution? Well, last time I mentioned that this occurs on a scale of 1 to, let's go ahead and put it there, 1 to 1,000 generations, okay? And that macroevolution usually requires greater than 10,000 generations, okay? So So, and obviously it's disturbing to know that there's, 1,001 to 9,999. What happens? Well, it's kind of fuzzy. But like I said last time, essentially what we need is time, and we need reproductive isolation. So what happens basically is you have a population that gets separated into two. Whether you have some geological feature like a mountain range or an ocean or water body that separates the population, or whether you have like a founder effect where a population migrates away and establishes a new population on an island or something. Either way, what you've done is you've eliminated the transfer of genes or the exchange of genes between two populations. And when that happens, basically, natural selection, sexual selection, genetic drift, mutation, horizontal gene transfer that happens in one population does not happen in the other population. So you have a divergence of experience that happens. And so time and reproductive isolation. So I want to finish this up by describing, let's see here. I'll open up here another file. Okay. Yeah. So, this whole concept, the term that we use, that we're going to talk about on Tuesday, we'll spend all of Tuesday on this kind of idea, is this idea of what's called allopatric species. Okay, so what allopatric speciation means, the term allo means separate, and patric means parents. So allopatric means the population is separated from its parents, or the parent population is separated into two subpopulations. So we have population of species A here on the top, and some event separates them into two in the allopatric speciation. In the case of vicariance, what vicariance means is vicariance is a geographic barrier, like a mountain range or a glaciation or a sea level change, that results basically in a parent population being divided into two. So if, say for example, a glacier intruded between two populations so that they no longer reproduce or exchange alleles, these individuals are reproducing among themselves, and any mutation or selection or drift or gene transfer that happens here does not get shared over here. And given enough time, even if these two populations were to come back together again, they might not reproduce. If there's enough change that happens, they may not recognize each other or be able to reproduce back with their closely related population. And that's an example of vicariance. Vicariance specifically is a geographic barrier of some kind. Mountains, glaciers, sea level change, rivers, river valleys like the Grand Canyon. Those are examples of vicariance. Got it? This will be the last thing here. Or you can have what we saw before, which was the founder effect, or population bottleneck, result basically in a founder species. So this is called dispersal. Dispersal or founder species. those are the two mechanisms for allopatric speciation. Either way, what happens is you have a separation of a parent population. And that's what allopatric literally means. Separate the parent population, or separate parents. Okay, so on Tuesday of next week, we're going to go over mechanisms that would result in, even if these populations were to reconvene, what would cause them to no longer be the same species, or basically, what leads to speciation. Okay, sound good? All right, let's take a 10-minute break. We have, it's not so bad today. We're looking at sponges.